It's a very proud moment for everyone present here and yes, for our college to have such a personality and dignity. Dr. Yogesh Mishra, Sir, Assistant Professor of Botany, Center of Advanced Studies in BHU. He has achieved the highest milestone in research development, the highest milestone in research development and academic at Banaras Hindu University as well as Max Planck Institute of Germany. We are honored to have you as our guest of honor. A big hand for him. Along with them, we have our blessed principal sir. Now I would request our principal sir, please come to the stage. And here I would request to the volunteers, the students of our college to come on the stage and sing the welcoming song.
ଟିକେ ମଝିକୁ ରଖିଲେ ହେବନି କି ଏପଟେ ସାଇଡ଼କୁ ହେଇଯାଇଛି ଟିକେ ଆଗକୁ ନେଇକି ତାଙ୍କ ଦେଖାଯାଉନି ଟିକେ ବୁଲାନ୍ତୁ
thanks to all the students for this beautiful moment. And I would like to welcome our HOD sir, Gautam Charan Das sir, and Himanshu Sekhar Mahapatra sir. Now I would like to request our principal sir to deliver some priceless lines to entertain and enlighten this intellectual gathering. Respected sir, please come to the podium. Please welcome. Thank you.
Very good morning to one and all present here. A warm welcome to all of you. I am feeling extremely honored to welcome all the distinguished delegates, guests, dignitaries, and research scholars to the inaugural session of two-day national seminar organized by Department of Botany. We are so delighted to have with us today our chief guest, Professor Ramchandra Mohanty, former professor and head PG Department of Botany, Utkal University. Guest of honor, Professor K. Vijay Kumar, former chairman, CSTT, New Delhi, Ministry of HRD, Government of India. Guest of honor, Professor Kunja Bihari Sakpati, former professor and head, PG Department of Botany, Utkal University. Guest of honor and keynote speaker of the seminar, Dr. Jogesh Mishra, Assistant Professor, Department of Botany, Banaras Hindu University. Coordinator UGC, Dr. Prabhakar Malik, all the members of organizing committee, all the faculty members of the department of the institution, former faculty members, paper presenters, and participants from the leading educational institutions of the state, and my esteemed teacher and ex-principal, Professor Nandakishore Parida, Professor Sheikh Samsur, Honorable Member, Governing Body, and ladies and gentlemen. Kendrapara Autonomous College is one of the premier autonomous colleges of the state of Odisha, the biggest educational institution in the non-government sector with NAAC re-accredited A grade. With the opening of new PG courses, this institution cherishes the desire to excel in research and generation to innovative ideas and to develop the teaching learning atmosphere for the welfare of the students and the society as a whole. To ignite and inspire and enrich the students, the institute organizes various seminars, workshops, and conferences at state, national, and international level every year. A seminar entitled "The Current Trends in Plant Science" is a burning topic on the exploration of recent advancements in the different field of plant science. Plants are indispensable for survival of all forms of life on the earth. Plant science is the multidisciplinary study of plants from the molecular workings of a plant cell to plants' roles in ecosystem functions. I hope that the seminar will provide comprehensive details of present day research and advanced technology in the field of plant science. I would like to congratulate the Department of Botany, its faculty members, for conducting such a seminar on the topic that is the need of the hour to be discussed. I wish all the very best to the organizing committee and hope this seminar a grand success. Thank you.
Now I'd like to welcome our HOD sir Professor Gautam Kumar Das sir will welcome us to the chief guest. Please, sir.
You welcome to all of you. Hello. A hearty welcome to all of you, esteemed Chairperson Patton and our beloved Principal, Professor Gopal Chandra Behra, Sir, revered Chief Guest, former HOD of Botany, Utkal University, Professor Ramachandra Mohanty, Sir, distinguished Guest of Honor, Chairman CSTT New Delhi, Professor K. Vijay Kumar Sir, Guest of Honor, Professor K.V. Kunjabihari Satpathy, former Head of the Department of Botany, Utkal University, and our keynote speaker and Guest of Honor, Professor Jogesh Mishra, Assistant Professor, Banaras Hindu University, and all the dignitaries present here, respected Samsur sir, Nanda sir, Ajay sir, and all the participants and students present here. It is my great pleasure, privilege to introduce the guest of honor. And introduction of the guest. Sir, Dr. Ramachandra Mohanty has served Utkal University as professor and head in Department of Botany. He has more than 37 years of research and teaching experience. He has been serving the cause of science in a wide spectrum of roles: educator, researcher, administrator, author, editor of books, popularization of science, promoter of talent, and as a thinker and planner. He guided 50 PhD and DSc scholars and many MPhil students in aquatic, terrestrial, microbiological and eco-toxicological areas of environmental biology in particular and plant science in general. He has published over 150 research papers and has written and edited several books. He worked out 9 major research schemes funded by agencies like MOEF, Government of India, DST, UGC, CSIR, CDA, ICAR. His inputs to the functioning of educational institutes, learned societies, cultural organizations, and professional agencies are a regime. Apart from teaching and research, Professor Mohanty has served Utkal University in various administrative capacities such as Registered Director, ASC Director, CDC, Chief of Employment Bureaus, and Head of the Department Botany. He has joined the Center of Excellence, Environment, Public Health, and Climate Change, Utkal University as the Distinguished Honorary Professor. He has served the state as a member SCSC for 2 terms. He received the prestigious Lifetime Achievements Award from Microbiological Society of India in 2021. His learning process never ended up and he got training from IISc Bangalore, TR Parsons Laboratory, Vancouver, Canada, etc. He also visited many countries like Canada, USA, USSR, UAE, the Netherlands, Thailand, Japan, UK and Germany in different academic assignments. Sir, we are fortunate to get a glimpse of knowledge from you in this august gathering. Professor K. Vijay Kumar, who was born on 19 October 1951 at Berhampur. Dr. K. Vijay Kumar completed his school and college education of Paralakhemudi. He topped in his MSc Botany from Berhampur University in 1972 and he awarded gold medal. He got his PhD degree from the same university in 1984. He has 70 publications to his credit in national and international journals. 13 PhD awardees and scores of MPhil students to his credit. Besides his research interest being physiology and biochemistry and environmental toxicology. He was a member of national and international learned societies like Scandinavian Society of Plant Physiology and Japanese Society of Plant Physiology. He also has 3 books as authors and 51 volumes of scientific glossary as editor to his credit. He entered Odisha Education Service in 1973 through BSc and served in FM College, Bangalore, Baleshwar, Kalikot College, Autonomous, Barampur, Revensa College, Kotak. He was selected as professor in 1992 through open selection and also acted as principal, Kalikot Autonomous College, Barampur. Coordinated IGNOR for more than 5 years and also established the Department of Biotechnology and the relevant laboratory in Kalikot Autonomous College. He was selected as the chairman, Commission of Scientific and Technical Terminology, CSTT, Ministry of HRD, Department of Higher Education, Government of India through All India selection, and also headed 2 more national level institutions, CST Delhi and KSC Agra under Ministry of Human Resource Development as director. Was a part of delegation of Ministry of External Affairs, Ministry of Home Affairs, besides Ministry of HRD to other countries and visited USA, UK, Mauritius and Sri Lanka. Others are the outstanding young scientists in 1991 by Indian Junior Chamber. Got Rajiv Gandhi Sadhana Award and Scientist for Excellence Award 2007 by Society for Plant Research in 2007. He was also associated with Rotary International. Sir, we are fortunate to get a glimpse of knowledge from you in this August gathering. Dr. Kendrapara Sarpati, Emeritus Professor of Botany of Centurion, University of Technology and Management, Bhubaneswar, and former head of the Department of Botany, Utkal University, Bhubaneswar. Had his early education in the University High School, Utkal University campus, Banibara, Bhubaneswar. After graduating in 1976 with a first class honors in botany with distinction from Regional College of Education, Bhubaneswar, he studied in the postgraduate department of botany, Utkal University, where he obtained MSc degree in first class with specialization in microbiology in 1978. He was awarded with MPhil, PhD and DSc degree from Utkal University in the year 1985, 1991, 2007 respectively. He is known in the state of Odisha as a person with expertise in plant taxonomy and his contribution to floristic study on medicinal and aromatic plants, in particular indeed phenomenal. Besides, he has been actively engaged in detoxification of municipal sewage, industrial effluents and mines wastewater through phytoremediation employing azolla and its floating associates. To date, Dr. Sarpati has supervised 21 students in their PhD research work in the field of botany, zoology, environmental science, microbiology and life science. 25 students in MPhil and 1 student in MTech and 48 students in their MSc dissertation work. 12 PhD students are still continuing their research work under his supervision. To his credit, he has published 319 numbers of research papers in national and international journals. In books chapter as well as conference proceeding volumes. His books on herbal cure of diabetes and azolla as soil fertilizer and waste disposal, published in 3 international languages, has been acclaimed worldwide. Recently, he has been felicitated by the Centurion University of Technology and Management for his 4 numbers of patents published and approved by the Government of Australia, Japan, South Africa and Germany during 2020, 21, 22 and 23 respectively. Dr. Sarpati was successfully executed 7 major research projects sponsored by ETC, OBB and ICAR and 13 consultancy projects on biodiversity, study funded by several industrial houses including NALCO, MCCL, Bhushan Steel Limited, NI, Hisa Steel, Maithan, Osil, etc. Professor Satpathy worked on plant biodiversity, inventories, qualitative and quantitative assessment of flora of Hemgiri of Sundargarh district, Sukinda Valley of Jajpur district, Telkoi and Borbil of Keonjhar district. He had extensively studied the ethnobiology of tribal rich districts of Odisha and reported over 1200 ethnomedical plant species and their uses in primary health care. He efforts to develop a protocol for production and utilization of azolla biofertilizer in rice cultivation and cooperation among the farmers in Odisha in enormous. Besides, he has been nationally honored when he was selected and received the Young Botanist Award for the best research paper by the Indian Botanical Society in 1984. Dr. Satpathy was the recipient of the prestigious International Ethnomedicine Research Award for the year 2014. Considering his achievements and outstanding contribution in plant taxonomy, the Odisha Botanical Society awarded him the Dr. Gopinath Panigrahi Memorial Award for Plant Taxonomy in 2015. He has been elected as the Secretary of Odisha Botanical Society for the tenure 2012 to 2015. Odisha Botanical Society also conferred Dr. Satpathy the College of Basic Science and Humanities Award for significant contribution to botany. Recently, Dr. Satpathy has been selected as one of the members of the Empowered Committee on Ayush. Dr. Satpathy holds over 40 years of experience in teaching research in the laboratory as well as in the field and environmental consultancy. Sir, we are fortunate to get a glimpse of knowledge from you in this august gathering. Dr. Jogesh Mishra is currently working as Assistant Professor at Department of Botany, Banaras Hindu University. He has more than 15 years of teaching and research experience. He is an expert with molecular biology, physiology, genomics, proteomics, and metabolomics of cyanobacteria and plants. His area of interest includes unraveling the molecular mechanism of abiotic stress tolerance and signaling in cyanobacteria and plants using interdisciplinary approaches. He has about 40 research papers to his credit and many more to come. He was a postdoctoral research at Max Planck Institute for Plant Breeding Research, Department of Plant Development Biology, Cologne, Germany, and advisor to Professor Sava Kunz. He was also a researcher at Umeå Plant Science Center, a center of excellence, Department of Plant Physiology, Umeå University, Sweden. He is also a member of International Plant Proteomics Organization, INPPO, and International Society of Photosynthesis Research, ISPR. He visited many countries like China, UK, France, Germany, Denmark, Finland, Switzerland, Sweden, Norway, and other European countries in different academic assignments. He was also successfully completed many projects funded by DPT, DSP, DI, and UGC. Today we are excited to get a chance to interact with such a young, dynamic, and vibrant academician. Thank you, sir. Thank you.
Thanks to Professor Gautam Charan Das sir for addressing the guests. Now I would like to invite our beloved sir, Himanshu Shekhar Mahapatra, lecturer in botany and joint organizing secretary for presentation of the theme of the seminar. Please come, sir.
सेवा से पढ़
समस्त नमस्कार. Head of the institutions and chairman of the meeting, esteemed principal sir, honorable chief guest of the occasion, Professor Ramachandra Mandi sir, respected guest of honor, Professor Vijay Kumar sir, honorable guest of honor, Professor Kunjabi Satpathy sir, esteemed guest of the honor, keynote speaker of the occasion, Dr. Jogesh Mishra sir, Honorable HOD Dr. Gautam Charan Das, respected UGC coordinator Dr. Prabhakar Malik sir, and all the dignitaries and the participants present here. It's my great privilege to communicate the theme of seminar on this occasion. As you know, the plants are essential for existence of all forms of life. Because the plants have capacity to photosynthesize, the plants supply water food material to us. So we directly and indirectly depend upon the plants. The plant science explore all aspects of plant life, including their practical and economic utility to the society. With the advancements of science and technology, many disciplines of plant science such as biochemistry, molecular biology, cell biology, genetics, genomics, epigenomics, proteomics, synthetic biology, stress physiology, molecular taxonomy, biotechnology, agriculture, and many more have been originated as separate branches under the umbrella of plant science to cater the need of the subject. In all branches of plant science, advanced parallel research is going on present day. Specifically, the plant biochemistry combines chemistry, physiology, and biology to investigate chemistry of living systems. Cell biology concerned with structural and functional aspect of cell in one of the emerging trends of the day. Molecular biology concerned with structure and interaction of cellular molecules, especially nucleic proteins to understand life. Genetics is the study of hereditary variation, as you know, but in present day, plant genetics study advances specifically based on the genetics of Neurospora and Arabis thaliana. Genomics and epigenomics are two most developing area of plant science that helps us to understand organization and function of the genome. Proteomics is the large-scale study of the proteins or proteins, which is the current evolving fields of plant science. Synthetic biology gaining importance in research present day involving the certain fully or operational biological systems from smallest molecules including DNA, protein and other molecules. Work on stress physiology nowadays help us to understand how plants adapt with the different prevailing conditions at biochemical and molecular levels. Plant biotechnology is one of the current exploring branch of plant science, having significant contribution in production of plant secondary metabolites, hybrids, and as well as rapid propagation of plants or clonal propagation. Molecular taxonomy is playing significant role in solving taxonomic disparity and help to find out evolutionary history of the species. Especially the integration of biochemistry, molecular biology, genetics, next generation sequencing, genome editing, and cell biology has paramount impact on plant science in recent years. The exploration of recent advancement in the different field of plant science for this seminar will provide comprehensive details of present day research and technology used. So this is all about the seminar, the theme and the sub-themes of the seminar. Thank you.
Thanks to Himanshu Sekhar Maharaj sir for the presentation. Now I would like to welcome the chief guest. First of all, proceeding the meeting program, we are very fortunate to have former professor, head PG Department of Botany with Kendrapara University as the chief guest. The legal agent, Professor Ramchandra Mohanty sir. Please sir come to this podium and deliver some priceless thanks to this seminar. Please, sit down sir.
Honorable President of this conversation, Professor Gopal Behra, Principal, Executive Principal of this great college, Kendrapara College, which I like very much. About 40 years before, I visited your college. At that time it attracted It was so nice. So I am first of all must be praising this college and these members, all these members, faculties and the students. After a long time I am meeting my old friend Professor K. Vijay Kumar. About say again 30 years, more than that. 30, maybe 10 years. No, not 10. It is 13 years. 13 years. 13 I was for half an hour or 1 hour. But I am talking about your events about 30 years. So I am extremely happy that my old friend is also here. My student, who is also a known scholar in taxonomy, Professor K.V. Satpathy, Gunjavia Satpathy, who is probably in this case is the pride as great scientist in taxonomy, doing much work, and he has got so many students at different places. He is gracing as the guest of honor, just like Professor K.V. Satpathy. Professor Yogesh Mishra, whom I do not know much, but I have long connection with BHU. When Professor Y.S.R.K. Sharma, I know, Professor Talab Sai, Professor H.T. Kumar, Professor Ambas, these are persons, and my close mates are Professor L.C. Rai, Professor B.R. Chaudhary, these friends are there. Now they are all retired, just like me. I am a retired person. And Professor Yogesh Mishra is doing wonderful work in molecular biology. I am really happy to know that. When I was there for some time in Indian Institute of Science, Bangalore, I had that was cell biology and microbiology. Professor T. Ramakrishnan was the chairman of that department. I enjoyed the topics so much there and stayed in Center of Ecology for again 2 to 3 months. So Indian Institute of Science, Banaras Hindu University, Madras University of Madras, University of MS University, Baroda. These are the centers from which I have got some knowledge. I was also there in Vancouver with Professor P.R. Parsons and did good work publishing papers. Professor Malik, Professor Das, Professor Himanshu, members of the faculty, members of the delegates, members of the teaching staff from different colleges and their students. Kendrapara would have been still more famous if it would be there in some other state like urban state like Maharashtra or Gujarat. Unfortunately, it is not the situation now. Do you know why? It's not because of our esteemed Lord Baldev Ji, but it is because of the Vitarkanika mangrove patch, which is very unique in the globe. Even in Sundarbans, we don't find such a beautiful diversity. There only it is one species, Ericeira, if I am correct. But here you find what not different types of mangroves. Unfortunately, because we are in Odisha, this is not being highlighted as it deserves. I'm sorry about that. Kendrapara is also famous for another thing, that is the Panika king. You do not know much about that, but he is the first person who did scientific work, that he tried to connect all the channels, all the water bodies that are prevalent in this district now, Kendrapara district. This is also famous for maritime activities. So, in many ways, Kendrapara is famous. And we like this district very much, very much. Now coming to the topic, I will not speak much because in my opinion, this is not having much relevance to our young minds, young students. When I speak on foreign trends, like say gene sequencing, DNA sequencing, or nucleotide sequencing, how many of them would be understanding that? So also the clone formation, if the same meristem, how clones are not monopoly of all biology people now. Plants also have clones, stem meristems, root meristems, leaf meristems. This could be cloned very much. So, the new cyanobacteria, Professor Rupesh Mishra will be agreeing with me. Now, this is a modern field of research that new cyanobacteria are coming up. People have tried to see the DNA composition is different. So also the poisonous viruses. Poisonous viruses we have been rejecting as microbiologists. I must say that. It is light strain, it is not lysogenic, and people have been rejecting this earlier. Nowadays, people think about the poisonous virus to be utilized for some medicinal help. So this way, if I go on speaking on modern topics, it's a long list, it will be a long list, but I do not want to speak on this aspect. What I will be speaking then, I will be talking on how knowledge is important for us. Whatever you are getting, my dear students, from your books, from your teachers, from the internet, wherever. These are all information, simply information. But knowledge will start when information is gained. That is the beginning point. You complete all the information. Just like today, I am not prepared at all. Extempore I will speak. And all the bookish knowledge or knowledge from teachers are ended there and knowledge starts there from. Which I wrote maybe last year in our Odisha Botanical Society annual meeting, that souvenir, that how people are imagining in Chirakas what should be the future of this country, what should be the future of this people, and what is our contribution as botanists. So that way, that is the knowledge which we get from different places, but mostly from our epics. I am remembering my childhood days. I have come from village, and there in my village they have the pleasure of doing this Ram Leela. And whenever the evening comes, we ask your mother that give us some kind of anything that you give. We will eat and go there. because the drums are beating now. The stage will begin, and you must be having some experience on that also. So what you are thinking, what you are enjoying there, it is all fun and entertainment. How Ram killed Ravan, how Surpanakha lost her nose, how Kumbhakarna has hibernated sleep. These things, how Ravan took away Sita. These are the important titles, selected titles. Just like nowadays, you study on selected titles. Similarly, you go there on selected occasions. So also Krishna Lila. Krishna Lila, you how Krishna has stolen all the saris from these gopis near the Yamuna river. That must be creating interest in you. How the Kalina was there in Kalanti Sarovar, and how Krishna jumped on, danced on the wood of this Kalina. These are certain titles where you must be interested in. But this, as I said, this is limited to fun and entertainment, nothing else. So what is the knowledge then? Just like our information in botany or any subject ends, and then knowledge starts, the information ends and knowledge starts. Similarly, if you study Mahabharata, all these episodes, you complete, these are all information. Now have become story to our children, history to us. But what is the knowledge? Knowledge is there in Bhagavad Gita. That you have karma yoga, you have bhakti yoga, you know how Arjuna was disturbed in war field in Kurukshetra. And Bhagavan Sri Krishna gave him the lesson, gave up him the knowledge that you do your karma. Karma yoga will bring you mukti or moksha. And not only karma, it is the bhakti yoga, which is still much better. Whatever sannyas yoga, sannyas is also yoga. Sannyas yoga is giving pain to your body. That is not good so much. Krishna says all these. So these things are there in Mahabharata. Similarly in Ramayana. Ramayana you study Yoga Vasishtha Ramayana. Vasishtha tells Ram that if you want to live merrily in this world, if you forget all your sorrows, then you do certain practices of yoga, so that you will be enlightened, you will have the moksha. So these are the knowledge. So knowledge you don't get from Ramayana or Mahabharata. Knowledge you get from Bhagavad Gita or Yoga Vasishtha Ramayana. You know, during these 2.5 years of Corona, I left botany completely, and I tried to translate Yoga Vasishtha Ramayana from Sanskrit. About 2.5 years, I wrote 400 pages. This is going to be published shortly. Yoga Vasishtha Ramayana. You have a glimpse on that. If at all you want to live happily in this world. It is a misnomer that people stay in family with family in this world cannot reach moksha or sannyas flow. feel that he can have this mukta or enlightenment. So, similarly, that was my point, only point, that you study your books for examination point of view. You study for gaining information. You obey your teachers. Visit libraries. Along with that, you try to have some. That is the reason why probably 2020 NEP, you know that National Education Policy, July 2020, we have accepted, our cabinet, central cabinet has accepted that. And there you have the extra credit. Now this cross the credit system is introduced for it, is it not? So UGC person is there, so he will agree with me that you complete all the credits of botany. Along with that, you have to study some other topic. Just like the gaining knowledge, interdisciplinary, yes, interdisciplinary, that is the emphasis to be given. Okay? So with this, I don't want to bore you any longer. Thank you very much. Thank you for asking me to come to this great college and speak to you from my heart. Thank you very much.
Thank you so much, Professor Ramchandra Mandi, for this beautiful speech. The information aims the knowledge stack, such an incredible line for all the students here. Now I'd like to welcome the guest of honor, Professor K. Vijay Kumar, former Chairman, CSTT New Delhi, Ministry of HRD, Government of India. Please, sir.
Good morning, everyone. I should start good morning. It is still 11:29, so it is still good morning. And I must address, but the chief patron, who is the president, is missing. So in his absence, I address him, Professor Gopal Behra. And today's chief guest, my senior friend, he is one batch above me, Professor R.C. Mahanti, and the UGC coordinator, Professor Malik, my junior but a friend, Professor Kunja Satpathy, and I must say, the Atiti Devo, our guest, Professor, I can call Professor Yogesh Mishra. Here it is Mishra, you are Mishra. So whatever it is, Mishra G, right? And my student, Gautam, and Himanshu, who is also missing. I know that. He was in college. And this sweet girl, I don't know her name. And from the audience, I must address one or two, but not all. I cannot remember, I cannot say. Madam Mohanty, good morning, madam, and Dr. Gopal. And Prabhakam. Then who else is left? Rabindra. He is and Prasanna, all this, and my student Suchasmita, and I must not forget to mention the HOD of Prevention, Durga. Sorry. Now, I mentioned all the names. There is a region also. I will come to that. Now, when I look at the topic, this is the most favorite topic of most of the seminars. Everywhere people say, what are the recent trends? So Rambabu, me, we are misfit to the recent trends. We are fit to the old trends. So I will try to put, this is what I have brought. I have brought this one, but I am not using it because so many years I am accustomed to speak extempore when these gadgets are not there. So I am more comfortable with that, and slides is okay, but not totally okay. Speaking from mind is, from heart is more appealing. So whatever it is. So my field of work, before coming to that, I will link the current trends and the old one. And I will link Gopal Chaudhary also to this. I will come to that. My first question to the audience is, when you were young, young means you were a school kid, children, did you want to become an adult faster? Yes or no? टेंथ में जाऊंगी, कैसे मैं ज्यादा बड़ा हो जाऊंगा और कॉलेज में जाऊंगा, फ्रीडम मिलेगा और other things are there, the Valentine days and other things. All these attractions are there in man. And once you cross that age, you go to the youth. After you cross the youth, what is that? That after youth, what is coming? Middle age. After middle age, old age. So anyone wants to become old? Yes or no? No, no one wants to become grey hair. Everyone wants to remain young. Okay, there are various reasons for that. You look old, all your organs go down, and you gradually lose everything. A few minutes back I was asking Dr. Mishra, what about Professor S.V. Kumar, the famous psychologist from Banaras University. He said, sir, वो ठीक है, लेकिन कभी कभार हेल्थ सेंटर में मेडिसिन लेते वक्त मिलते हैं. So that is the importance of old age. Where we meet, we also meet that way in such seminars or in clinics. If I go to Apollo Hospital or AIMS or CARE or anywhere, I meet some friends there who have come for their regular checkup or whatever checkup, diabetes or heart or whatever it is. So no one wants to become old. Everyone wants to become young, either adolescent or child. Okay. So when we do not want to become old, so we have to arrest the factors that lead to the aging. So my field of research, when I started, was aging, senescence. I don't know how many of you know the word senescence, because senescence is rather a technical compared to aging. But aging and senescence are two different words. Now, where from this concept of aging to my mind came, or why did I start this? Now, this senescence research, that means leaf senescence. What color do leaves get after they become old? बताइए बच्चों, इनके लिए नहीं. Leaves का कलर क्या होता है? What happens to the color of leaf? येलो, येलो के बाद the leaf falls and new leaves come out. Different types of senescence are there. That is not the point of discussion here. So, plant senesce. Do we senesce? Do we become old? But aging is different. I will tell you the basic difference between senescence and aging. Aging is growing older. Senescence is the cell stops dividing, both in humans and plants everywhere. Cell stops dividing. In aging, the cell gets destroyed, but in senescence, age stops, the cell stops dividing. So my work was how to stop the senescence. So what we did, we used chemicals so that leaves and plants will remain green for a longer time and for a better crop yield, leaves become green. Now, the pioneers in this work, younger people and those who do not know must know, this was started by Professor Radhadhar Mishra, the professor in Revancha College. Then it was taken over by Dinabandhu Mishra, and later on we had taken up the mantle. So what we were studying? We were studying what happens to chlorophyll, what happens to proteins, what happens to nucleic acids, and how those changes can be stopped. Just this much was our physiological and biochemical work. But now what? Now let me come to current trends. I was sitting there and I was asking Dr. Gopal that what is your field of work? He said, sir, paroxysm, right? And I link myself to that. I have worked on two enzymes when I was working. It was peroxidase and catalase. These two are peroxisomal enzymes. What I was doing? I was extracting the enzyme and analyzing its activity. And what you are doing? You are finding the genetic markers, molecular markers, the genes that are coding. These things you are doing. That is the current. Now, we were studying proteins, nucleic acids. What are they? What they are in the leaves or anywhere. Now, what is Dr. Mishra doing? He is working in proteomics and genomics and transcriptomics, I think. Transcriptomics. Now, some students may not understand what is transcriptomics, what is proteomics, what is phenologics, what are all these things. These are the trends that are coming up. Now, we were locked up at a stage which was biochemical. We were studying simply biochemical. We were not studying the molecular aspects or nucleic acid or nucleotide or genes. These things we are not studying. Whenever they have been studied in an indirect way, not in a direct way. Not what the current researchers are doing. Now, Dr. Barik has done on tissue culture. He must have done some fingerprinting algorithm. Whatever genes he has isolated, PCR, everything. When I started biochemistry laboratory in Kaligod College in 2000, there was nothing. Now, no techniques were known. Nothing was, how many years back? 23 years back. At that time, it was very difficult to do something. Now, everything is done in an automated way. Now, sequencer, we used to show a diagram. At that time, we had ordered a PCR, which was only not real time, not any advanced, no reverse transcriptase, nothing, simply a PCR for analysis. So gradually things have developed and it will develop on and on. So I understood that no one of you wants to become old. Everyone wants to remain young. Where you have to attack, you are not a plant, you are an animal. So what are the things you have to stop? You have to stop mitochondria changing. That is the hallmark of aging. And senescence if it occurs. Acha, from common sense can you tell me? Senescence I said cells stop dividing. Is it useful for cancer or harmful for cancer? Cells stop dividing if you are senescing. Cells are senescing. The cell stops division. Is it useful for cancer or harmful? What is cancer? Definitely. It is beneficial because the cells won't divide. In cancer, the cells divide. If senescence occurs, then cells won't divide. But recent researchers have found that accumulation of more and more senescent cells causes also cancer. Hypothesis, counter hypothesis. Senescence is useful, senescence is harmful. Both ways they deal it. So, it is all a complicated thing, and you young minds with current trends, now everything is available. The other day we were discussing, in our times you had to do everything. Now, if you do not have a machine, you can give your material to someone, he will analyze it and give you. Only thing that is needed is you must have money. Now, research is linked with money. Now, I can very well understand the position of Kendrapara College or for that matter any other college, because in colleges it is very difficult to do research because modern facilities cannot be obtained in colleges. So you must do research like our Professor Kunja Satpathy, who has come in ethnobotany, and whatever our traditions are there, you can carry out research in that. If you want to go in molecular level, most unfortunate thing is, whenever I ask a student of botany or biotechnology, what you want to become, and what is the answer? I want to cut what? DNA. I want to cut DNA and join DNA. Except in that, nothing. Oh, everyone wants to go for modern techniques. Arey bhai, the best thing is, when everyone is running after something, you should run after some other thing where things are easily available for you and achieve wonders. Now, you see what Professor Satpathy has achieved. And forest research, conservation, these things are also important. So don't think that taking out anything, cutting it, analyzing it is only important. You can go anywhere you want. You can do any type of research. But research is important now for choosing a career, either as a teacher or as a researcher. It is a must. Now, you have to move out, you have to work hard. I will tell you the example of one of my students, my first batch biotechnology in Kalkut College. There was a student, +2, third division, BSc, second class. He somehow got into the biotech. He was not able to speak good, English, but he worked hard. And I won't name him, not the institute where he is, and he is now in a leading research institute in Delhi, genomics. So that is what is the reward for hard work. Teachers and parents always say, work hard, work hard. It is not without any intention. The intention is that if you work hard, you will get the reward. Professor Mahathir spoke about religion, Mahabharata, whatever it is, everywhere you should work hard and don't expect the result. Result will almost come to you, back to you at any time. Now, I am happy. This is for the second time that I am visiting this college. I am happy that I have been invited. Even if I had given a PowerPoint presentation, it could have been a technical paper, but not based on my research, but based on the material available. Because I am away from research since many years. And in the meantime, as Professor Mishra told, in the last 10-15 years, there is enormous rise in the work and also the talented students, those who are coming back to India from foreign countries. So this is the time to improve yourself. Don't keep in mind, again as I said that cutting DNA, similarly people think that I can do research only if I could go to USA or UK or Germany as XYZ has gone. That is not the thing. Anywhere where you are, there also you can achieve things and you can do research. And it is more difficult for people in this college or any such college to do research work. But if you want to go ahead with the current trends, you have to go with what is available to you and what you can get. So my dear students and colleagues, those who are in research, please try hard to achieve something in life and that will take you to modern or current trends. The current trends today are totally different from current trends after 5 years. So trends change. You must update yourself. Now, I learned computer in 2020. Dr. Mishra said when I got into MSc, he was not born. And I can't speak about Gopal, he must be somewhere. But even when I passed MSc, he must not have been born. So good timers, we are out, we are going out, we will be meeting in health center and some place or other, some marriage or somewhere we may meet old friends, but it is the younger generation which has to carry the baton from your seniors now. They are all your seniors now for the students I am taking, and you have to carry. Those who are in whatever field, let them continue in that and be stronger in that field. Don't say that I could have done this and I gone to MIT, had I gone to this university, that university. Don't think that way. I thank very much all my friends present here. I call them all the friends. And my students who are here, direct students, and my young colleagues, Dr. Mishra, Dr. Chaudhary, all of them. And I hope this two-day seminar will be a success. And it will be a success because we have invited such eminent current trend experts like Mishra to speak over here. And the traditional part of it, Professor Kundu Sampati will take over, and definitely this seminar will be a success. Thank you very much.
We are really grateful to have you in the stage. Thank you so much, sir, for this eminency. So don't we all work hard and get the reward. Thanks for a beautiful speech, sir. Now I'd like to welcome. It's a blessing to have such a living legend, our guest of honor, former Professor Kunja Bihari Saraswati, sir, PG Department of Botany, Utkal University. So please come.
Honorable President, Principal Kendrapara College, Professor Gopal Chandra Bihara, respected chief guest of this national seminar, and my esteemed teacher, Professor Ramachandra Mahanti, esteemed guest of honor, Professor K. Vijay Kumar sir, respected guest of honor, Dr. Jogesh Mishra, Honorable Head of the Department of Botany of this prestigious college, and convener of this national seminar, Dr. Gautam Charan Das, respected Joint Secretary, Professor Himanshu Shekhar Mahapatra, all the distinguished resource persons, delegates, researchers, teachers, and respected faculty members of the Department of Botany and other department of this college, and dear student participants, ladies and gentlemen. Madam Professor Mahanti and Prasanna sir, again I offer my pranam to Madam. At the outset, I wish to offer my deep sense of gratitude to the principal of this college, Professor Bihara, and all the staff members, staff and students of this department botany of this college for inviting me and giving an opportunity to share some of my experiences with the participants of this seminar titled Trends, the Current Trends in Plant Science. For your clarification, I am not a taxonomist, but in my student career, we did not have this specialization in taxonomy. I did my MPhil in plant physiology and biochemistry and did my PhD at CRI on the biofertilizer technology for enhancement of the rice production, and my BSc work is under the able guidance of Professor R.C. Mahanti, that is ecophysiology and biotechnology. But I am guiding students in taxonomy. I am happy to know that the themes and sub-themes of this national seminar includes both traditional as well as applied aspects of plant science research, especially diversity of lower plants, plant systematics, ethnobotany, microbiology, and biodiversity. The main purpose of my deliberation is to motivate the participants, especially the student participants, to explore and identify the diverse group of plants, including lower plants, available in their surroundings and their utility or application in human welfare. As you know, that plants have been intimately associated with human civilization since ancient time. Man, being the most intelligent, rational, and social amongst the animals, has learned to utilize the phytoresources or the plant resources of their neighborhood for heroic purposes, as medicinal plants. Besides exploiting plants to cater their bare needs of living, food, shelter and clothing. We all know that plant resources are being utilized by all of us in the form of food, fodder, fuel, fertilizer, fiber, medicine, beverages, etc. So we all are dependent on these plant resources for our basic needs. There may be up two categories of phytoresources utilized by us in our daily life. The number one, cultivated ones and the plants growing in wild. It is very easy to identify and explore the cultivated or domesticated plants, but difficult to explore and identify wild, uncommon or less known plant resources around us which are also having utility. Therefore, it is the need of the hour to explore and identify the less known wild plants or wild relatives of the domesticated or cultivated plants having utility for human welfare. This will enhance our knowledge on the occurrence, distribution and utility of these wild, less known or unknown valuable phytoresources of a particular region. For the benefit of this, because most of them are the participants are undergraduate students. For the benefit of these students, I wish to categorize phytoresources under various groups basing on their economic importance. The plant resources are found in the form of food and food adjuncts, fuel, fertilizer, edible oils, essential oils, medicines and beverages. If you start with food, it includes cereals, pulses, oilseeds, which are the source of carbohydrates, proteins and lipids. There are green vegetables and fruits provide necessary minerals and vitamins. Similarly, there are two categories of fruits, just like your cereals. You know this swa chawla is also utilized by the rural people. Similarly, many of the cereals and pulses are being used by the tribal and hill community for their as their food. And the same way, we know that only the fruits which are available in the market, and those are mango, banana, papaya, grapes, fruits and oranges, lemon, pineapple, jackfruit, coconut, etc., which provide the body with minerals, salts and vitamins. But there are some wild edible plants which are available in the forest area as well as in the rural area, which are also having the same property, and these are being used by the tribal people and the common people in the rural Orissa as food, as fruit. For example, I will give examples in Odia because you may not, the students cannot identify the plant. You know that the amla, then phira pori, chara pori, kontei pori, bara pori, bhui bara pori. So there are many others are there. But these are the wild edible foods which are being used by the tribal people and the hill community in the forest area. So they are also having same properties, same value. Similarly, leafy vegetables which are available in the market, we only know the groups of the amaranth, khada sagar, leutia sagar, we know this. And also moringa, sadhana sagar and bathua sagar, sorry, not palanga sagar. But there are many other leafy vegetables which are being used by the tribal people. More than 128 tribal peoples that these leafy vegetables are being used by the tribes of Orissa. Out of which I just mentioned 3-4 to which you are acquainted. Alternanza society, that is Madaranga, Tita Gahama, Sunsunya sagar, Ambilti sagar, Balbalwa sagar and Bathua sagar. So these are the wild plants
and many others are there which are being used by the tribal people as leafy vegetables. So we must explore more the more strength and species of this type of wild leafy vegetables, and that should be evaluated. So that is a current trend we'll discuss. Then the underground crops. We know only few which are being sold in the market. Stem tubers, we know the potato and yam, root tubers, sweet potato, radish and turnip, and similarly bulbs, onion and garlic. But there are some wild tubers which are very rich in the nutrients and also having pharmaceutical importance. For example, you know the tubers of Satavari, that is asparagus racemosus, bhuni kakaru, pyrolia tuberosa, paruo, that is another wild plant, wild haldi, vana haldi, and Parsukela, Jindivar Telumbe. So these are the plants which are being used by the tribal people. And we do not care this plant because we do not know the use of this plant. Similarly, food adjuncts. Learn to use the food adjuncts during the corona pandemic period. According to the World Health Organization, during that period, you should use condiments and spices, which are the source of immunity boosting properties. But we used to use some condiments or the food adjuncts, which are very popular, like ginger, turmeric, haladi, cloves, cinnamon, black pepper, cardamom, fenugreek, nutmeg, and asafoetida, which are some of the food adjuncts that enhance the taste, flavor, and appetite, as well as immunity against diseases. But during the corona period, we are advised to use the kadha. Take the kadha, which contains these condiments and some other materials which we do not care and do not use. So that Guruji, you must have heard about Giloy. Giloy is this Hindi name, and it is nothing but your Pinospora corifolia, Guruji. Guruji powder is very important, having immunity boosting properties. Jastimatu, it is uncommon. We do not use actually daily dishes. It is also having immunity boosting properties. Ashwagandha, then Satavari, Oida, and lemon. So these are the ingredients of the kadha, which are prescribed by both Ayush Government of India and also by World Health Organization. Then comes the food, the fodder. You know that we know only the grasses and the fodder, but the people of the tribal belt, that they use the fruits of many plants as fodder, and one plant I will mention on which I did my PhD work. Azolla pinnata, Azolla biofertilizer for rice production. So Azolla is a protein. We call it in Odia Tunidara. It is used as a crop for the poultry, for fishes, and for cattle. And that is a strong nitrogen fixing complex, having both pteridophyte and frugalin alga anabina azoli, which are capable of fixing atmospheric nitrogen and supply the nitrogenous nutrients to the foreign crop of the rice. Similarly, fuel, you know we know only firewoods use as fuel, but now you must have heard about biodiesels. So biodiesels are the fuel for the vehicles, and we know only Kendrapara, Baigava, Manji. But there are some other seeds are also used for the production of biodiesel. Koranja, Kusuma, then this Modoka indica tola, Mahula tola manji. So these are used as the source of biodiesel, fertilizers. Previously we know that Dhanicha is a leguminous plant which is used as a biofertilizer or green manure before the cultivation of rice. But there are many others have been discovered many species of the blue-green algae which are capable of fixing atmospheric nitrogen and are advised or are recommended for the rice cultivation to enhance the crop productivity. It can supply 30 to 60 kg nitrogen per hectare per year per crop. So that is why Azolla and blue-green algae, you must have heard about this student, the Anamina, Nosta, Skytonema, and this many others are there, this Limbaya. So these are the blue-green algae, alga, so which are having ability to fix atmospheric nitrogen can be recommended for the cultivation of rice to enhance its productivity. Fiber, the water and food, edible oils, mustard and groundnut, but the tribal people, they used to use some seeds as the source of their edible oil. Then the essential oils, so these are, Camarada, lemongrass and citronella. So these are the plants which are the source of essential oils, and these oils are used in perfumery industry and tobacco industry to enhance the flavor. Similarly, medicines, all the plants are having medicinal properties. You know that Indian system of medicine, the Indian system of medicine includes Ayurveda, Siddha, Unani. So all these Indian system of medicines are based on the plant materials. So according to World Health Organization, 85% of the Indian system of medicine is based on plant or their plant products. Similarly, this white man to refresh himself, but some tribal people, they use some beverages. The country wine like Handia, dead palm juice, that toddy, and solapa juice, so that is also used. So these are the soft drink used by the tribal people. Yes. Professor K. Vijay Kumar, he mentioned about the modern trends and traditional branches of botany. So we have tried to use this traditional knowledge and evaluate the authenticity of the medicinal plant which are being used by the tribal people. That is, folklore claims should be authenticated by phytochemical screening, this pharmacological evaluation, biological evaluation, and toxicological studies. So these are the modern trends to evaluate the authenticity of the medicinal plant claimed by the tribal people. Similarly, when I visited the Raupla steel plant effluent the site, and also the mine wastewater at Ariapadi and Nirate Ispat Nigam Limited. So we have tried some plants to decontaminate the wastewater, including mine wastewater, sewage, and also industrial effluent. We could find that there are some plants are growing in the water body receiving these wastewaters, like industrial effluent. So the canal or river, they are forayed with these effluents, and those stagnant water is rich in biodiversity, aquatic plants, floating plants, and some blue-green algae also. So the phytoremediation is a technology which is employed for decontamination of this wastewater, including sewage, mine wastewater, and industrial effluent by employing plants. So similarly, many plants are floating on the surface of the water body receiving the wastewater. So you can identify those plants and try with this wastewater to decontaminate this. So this is the modern trend. Yeah. Similarly, this solving the taxonomy problem. So there are some plants very nearer to each other. So to solve the problem, we can use the method of modern taxonomy, chemotaxonomy, molecular taxonomy. So to solve the taxonomic problem, we have to take the help of this modern technology or this modern trends in botany. So what is the necessary? We are not neglecting a branch of botany, which is the mother of plant science, that taxonomy, morphology and taxonomy. It is neglected, and the taxonomists in Odisha and in India are critically endangered. You cannot, because the students are not opting for this taxonomy, but to pursue any type of research, you have to take the material, that is a plant, if you cannot identify yourself and give the plant material to a taxonomy who does not know about this taxonomy and it identified, then that result will be erroneous. That is why the taxonomy is a must for all plant science students. It was, so what is the necessity of taxonomy? I just take 4-5 minutes. Taxonomy for identification of these plants. It is found that some plants are known by different names and different plants are known by same name, which create elements of confusion. So I will give two examples. One person from Gopam Ayurvedic College got PhD. He has taken two plants and named as Punarnava. But both the plants are different, belong to two distantly placed families. But he got PhD because they do not know the identity of that plant and did not take the help of any taxonomy. Similarly, another very renowned plant researcher, he published a paper in Elsevier Journal, and that plant he took from Kendrapara, but that plant is not available in Odisha. So that is the problem. We just follow the protocol and do many things. So that will create confusion and also the result will not be effective. Therefore, scientific naming of plants is required to know what by single correct valid name. I'll give one example. You know our mango, amber. So mango is having several names, vernacular names throughout across the world. But if you say Mangifera indica, which is the scientific name of mango, so everybody from any corner of the world, they know that it is mango. So that is why the scientific name is required. This method of naming is based on certain principles for the plant science students. It is based on certain principles governed by a set of rules printed in the form of a code, a book known as ICBN, International Code of Botanical Nomenclature. Now, in 2013, the name has been changed. It is ICN at Melbourne. The name has been changed. That is International Code of Nomenclature. For example, single plant is known by a number of names in Odisha. I will give one example. Guava. Guava in Odisha, in different parts of Odisha, is known as Pijuli, Piara, Chauria. But botanical name is Sidum guava. So a single plant is named differently. Custard apple you know, is a sitaphala, ramaphala, bodhiyala, apple. But that is known as Alola reticula. Similarly, you know this Singara, Cephalic, for the same plant, that is Nicanthus arborvitae. So during this corona period, the use of the leaf is very popular for emission of the temperature. Then this, you must have seen in the Rabi crop field, that is known as Bhui Kadambo. Bhui Kadambo, we call it in some parts, Bhui Kadambo, in Kendrapara it is known as Patopulia, and somewhere it is known as Batuaguli. The plant is Paranthus indicus. It is a medicinal plant included in Ayurveda. Similarly, a number of plants is known by a single name. I will give one example. One Ayurvedic drug known as Bodaristo, or sometimes it is called Badamuta. It is given to the children for. So this Boda is named for 4 plants. You know that Vajradanti, Dantadanti, which is prepared from the root of a plant, if you do not identify the proper plant, so that the pest and the medicine will not be effective. you know that Saida acuta, we call it Bajramuli. Then the Saida caudifolia, in Odia we call it Bisiripi. And Agotinal indicum, we call it Pedipedica. Another plant is known as Saida cordata, it is Bhumibandha. All the four plants are called Boda in Ayurveda. Then we have to evaluate the phytoconstituent of this plant which target a particular disease. The plant which having more amount of this phytoconstituent targeting that particular disease or targeting that particular situation. So we have to identify by employing the modern technology, that is modern trends or the current trends in botany. Another plant is not Samphapushpi, Samphapushpi Toda and this Gruta is available. So four plants are known as Samphapushpi. So suppose a pharmaceutical industry use one plant and another pharmaceutical industry use another plant. So the effectiveness will be different. So in Odisha, we call that Gandhi Pohoda, Canscoratica Samphapushpi. In South Kerala and Tamil Nadu, they call this Operagica, Clitoria ternacea as Samphapushpi. In North, they call it Convolvulus pulicaris as Samphapushpi. So that is that create confusion. So we have to, with the current scientists or this modern scientists, we have to evaluate the Dravya Guna, that is phytoconstituent, which is responsible for the preparation of that drug. Brahmi, in Odisha, we call Pacopa mani as Brahmi. But in North, they call Thalpudi as Brahmi. Central Asiatica, that is Mandukaparni in Sanskrit. But both the plants are having similar phytoconstituent, that is Brahmin and Bacopin, for which it is already recommended. Both the plants can be used for the same purpose, that is a memory enhancer. Rasna, you know that epiphytic plant Rasna. So there are more than 150 types of epiphytic plants are available in Orissa. It was identified by Dr. Sarat Chandra Mishra. So Rasna is Bodaka, which is used against arthritis and rheumatoid arthritis. So that is prepared out of the root of this plant. So there are four plants, they call Rasna. So again, it creates confusion. Similarly in Chirita, you know that in our early childhood, we used to use Talmenga. Talmenga is a syrup prepared by Bengal chemicals and used against this any type of gastric derangement. And we have two plants here in Odisha, Androgaphis guinimper, Androgaphis pedicurata and Androgaphis dicoides. Both the plants are used for the same purpose. But in North India, in temperate climate, Swastia thyraida is available. They are not found in Odisha. So it is found that all the ingredients which are responsible for that particular target purpose are found in all the three plants. Similarly, Bringaraj, Bringaraj is available for the luxury and growth of the hair. So in Odisha, we call it Bringaraj, that is Odelia calendulaceae having yellow flower, is the Bringaraj. But in other parts of the okay. So there are many plants which are having confusing identity. We have to identify by employing the modern technology, and that is the modern trends in plant science. The desired efficacy of the plant-based drug is often not achieved because of using a raw material with a confusing identity for the preparation of drug. Incorrect identification leading to use of a wrong plant is often responsible for deterioration in the quality of the drug and also the effectiveness of the medicine prepared, and at times even it leads to serious consequences. It will fatal if you. So last sentence I will tell you that you know that Chyawanprash, this was recommended first during this Corona period. It is composed of 30 plants. It is very difficult to identify these 30 plants. But the pharmaceutical industry, local pharmaceutical industry, they are preparing this Chyawanprash. All the pharmaceutical industry may not use all these 30 plants. Some plants might have been deleted. So the authenticity we can verified by morphologically, by taxonomy identification, or in silico analysis, or phytochemical screening, or pharmacological as well as biological evaluations by mouse model, or taking the living organisms. With I am sorry that I extended my talk. With this, I thank the participants, all the respected participants for their patience here. Thank you.
Thank you so much, sir. Now the time is coming. I would like to request all the fraternity release of souvenir. I will request to Professor R.C. Mohanty, sir, chief guest, and honorable dignitaries on the dais for release of souvenir. Thank you everyone, honorable guests on the dais. So now the time is coming. It's a very proud moment for everyone present here and yes, for our college to have such a personality and dignity. Dr. Yogesh Mishra sir, Assistant Professor of Botany, Center of Advanced Studies, BHU. He has achieved the highest milestone in research development and academics at Banaras Hindu University as well as Max Planck Institute, Germany. We are honored to have you as our guest of honor. A big hand for him. Please come here, sir. Thank you.
A very good afternoon to all the academic dignitaries on the dais, off the dais. Since I'm not familiar with most of them, I'm very sorry because so that's why I'm sorry because there are many great personalities sitting in the audience. Thanks organizing committee for giving me this opportunity to deliver a talk, technical advances in the plant science. And when I was contacted by to give a talk on that, so it was a big surprise. And I thought, how I am going to summarize the last 3 decades advances in the plant science? It was very tough. And since a lot of students are sitting here, before starting this thing, I can assure you the upcoming 10-20 years is going to belong to the agrobiotechnology. So please understand this thought, and if you are going to pursue a career with good amalgamation of basics as well as advanced knowledge, nobody on this planet can stop you to achieve the success. So with this word, let me start. So if I have to start, so if you look at the current thing that what is the virtuous cycle of science and societies shows how each advances of each other. As many of the earlier speakers talks about their research, that what they have been doing and what there have been expertise. So it's a very continuous process. Here you can see that, can you please go back? Yeah. So if you look at the discovery, scientists make discovery, integrate the discovery generated knowledge, and knowledge going to the society, and society develops the scientists. This is the cycle which has been growing, it is going, and it will be continued. It is depend on the situation and need of the hour. We are following this cycle. So we have to acknowledge the effort of each and every scientist, because science is a continuous journey, and we the scientists, we just make one step in that. So the baton will be passed on to the next level, and next level, next level. So we should never use the word that earlier people have not done. They have done the things by their own way. And you know that we are part of this cycle, and we are continuing it. And so if you look at this thing, so plants is one of the most fascinating and important group of organisms living on earth. They serve as conduct the energy to biosphere, provide food and save our knowledge. And here I put one of the diffraction image, and I guess most of the students are aware about the fact. This is the kind of a beginning where we solve the structure of the DNA, 1953. And if you look at, I will show you the timeline. That is the kind of development of the modern biology, how the people are starting looking deep into the nucleic acid at the structure level. So the two essential role of any new technology, that advances in the technology provide a mean to test the hypothesis, formulate and less complicated and less practice information. Technology provides information to generate fresh hypotheses, and here I put that, for example, the technology like X-ray diffraction help in testing the hypothesis generated from chemical analysis leading to the discovery of DNA and uncovered vast new field of the information. So that's why, that is the reason behind putting this part. So it was a very tough task for me, that how I am going to put all these recent advances as a keynote speaker in a title that recent advances in plant science. So I categorize my talks in the three categories. That if you look at the development of the plant sciences, it was the molecular biology which was going from 1974, because the first gene which was cloned was the photoliase in 1974, that was the Stanford University. So 1974 we know the cloning, but the boom is coming after the development of many new technology, sequencing technology and all these things. So I put that the technical advancements before the using the traditional approaches, and then the genome editing technology when the NGS platform come after the 2003 onward, how the things have changed, and the system biology approach like the large scale biology. So audience, I will try to compile my entire talks in the three parts. So here, if you look at the traditional approach, because people were much more fascinating that how to get the desired traits in the plants by using the classical breeding and using the cross-breeding approach. And of course, it achieved many success and is still continuing, but there was a limitation of the time because people want the response quick. In order to do that, the Arabidopsis thaliana developed as a model system. And it's my proud moment that one of the person who has been involved in developing of this model, that it belongs to the Max Planck Institute. Jepsen, I will talk about him later because he was the director of Max Planck Institute, Cologne, Germany, and who was the person who established that agrobacterial mediated transformation is possible in plant. So I will talk in a minute about that. Introduction of Arabidopsis thaliana as a model, agro-mediated transformation and tDNA binary vector system formation. How we have developed this vector and what was the technological evolution made by this introduction. So if you look at Arabidopsis thaliana, Arabidopsis thaliana was a study material in 1980s. Many people were doing research in 1980s using the Arabidopsis, this tiny plants. And for the students, you may you may feel that Arabidopsis thaliana is not found in India, but it does, because there is around 39 accession of Arabidopsis thaliana has been reported worldwide, and one of the Arabidopsis thaliana accession casts from Kashmir is found in India also. So this is for the general knowledge for the students, and it has been employed a lot. And the Arabidopsis reference, because when a lot of people were using, and they found that they are having almost all the important attributes which a model plant should have, like small size, quick generation times, ability to self-fertilize, ability to outcross, it's easy to handle. So it was in the scientific fraternity, and that's led to development of first complete genome sequence of Arabidopsis thaliana, and it was completed in 2001. 25th May is celebrated as the Arabidopsis Day worldwide. And when we have the complete genome sequence and all these things, then the next question is that can we transform this organism? Can we do the genetic manipulation in this plant or not? In order to do that, a lot of things has been done, and so here, because at the same time, people were, one of the scientists, this is Notru and Joseph Shell, that is, he was a person who was working in Belgium, in Ghent University. So he did a lot on the agrobacterium tumescens, and look at how the two things were growing. That he was the person who has given the word the agrobacterium tumescens, and he did a lot of changes in the plasmid so that it can be used for the manipulating the gene. And 2001 onward, he became the director of Max Planck Institute, Cologne, Germany. So we all know about the agrobacterium mediated genetic transformation. I am not going to take your time to explaining that how it does and what are the features. But for the students, like when I teach the MSc class, I just should introduce that how it do. So this is the TI plasmid, which is basically responsible for transferring the gene of interest, because there is a left border, right border, and arginine, cytokinin. These are the three important genes which is found, and the size is like 12 to 12 base pair. So this left border and right border things are very important. For the student, if you put anything in between, it will be easy to transfer. So this is how the plasmid came to know that it can induce the tumor in the plant, and it has the different reasons, open catamelism, virulence, and the different reasons. I'm not going to talk about it. But the important thing is that, can you do the next? So here, so one of the things which was established that using agrobacterium tumescens TI plasmid, you can put the desired trait into the plants. The mechanism is well known. I'm not going to, anybody can read in the books, especially for the students. The important thing is that this point is very important. This process is called as allogeneic recombination, does not depend on the sequence similarity. That is the key feature one should keep in the mind, that if you put anything between left and right border, it will be transferred. So that was the boon for the genetic engineers, that the desired trait can be put into the plant of interest. So that is the main point which I want to highlight. So the important thing is that since the TI plasmid is very big plasmid, it is very hard to handle. Then the scientists, especially led by the Jepsel, they try to put the desired characters on the two different plasmids. So that I'm not going to explain here, but the next slide please. So a lot of modification has been done in this TI plasmid. I would like to just keep certain things in your brain, that when we do the transformation, we want that our gene of interest should be transcribed. So we put the two origin of replication site, one for the bacteria and one for the plants, so that we can maintain the replication of gene of interest in the desired plant, and we put some selectable markers so that when we do the transformation, we can select the transformed colonies. So these manipulations were done, and that led to the development of the binary vector system, and that binary vector system, I would say, the first very important technical advancing things that given a tool to the scientists that you can do the gene manipulation by using the tDNA or binary TI plasmid based binary vector system. And this credit goes to Jepsen, and that paper published in Nature. And I'm happy that I didn't meet him, but whenever you go to Max Planck, people are talking about the Jepsen, and you might know the company Bias. This is very big company, seed company that was developed by him, and now the VIV Ghent is one of the leading plant science center in Belgium that is known and that is established by the Jepsen, who is the main person behind the development of the agrobacterial mediated transformation. So I would say that is the first breakthrough, the first technical advancement that scientists came across, especially on the plant transformation thing. And now the binary vector applied for a lot of plants, a lot of species for transformation. And right now people are using for transferring the liver birds also agrobacterial mediated system. So it means that is one of the biggest advancement has been happened in the area of plant biotechnology. So let my slide on, then I will proceed forward. Thank you. Can we move next? Next, please. हिमांशु थोड़ा सा लैपटॉप टेढ़ा कर दिए, देन इट विल वर्क। वो सेंसर उसका पा नहीं रहा, थोड़ा सा इसको टेढ़ा कर दीजिए, थोड़ा सा आगे कर दीजिए। हाँ, so that's all. So here you can see this is binary vector system. That is the one of the greatest development, and it started from the original TI plasmid. Here you can see the left border, right border, and you are having the multiple cloning site. You are having the recruiter and promoter, two origin of site, and the helper plasmid which introduced. So that is one of the technical advancement, and people are using for the different plants for the putting the desirable traits in the different plant varieties. Next please. So this is next please. Now coming back to genome editing, because nowadays it's a very fancy subject to talk about the genome editing. And for those, because you know there was a huge campaign across the worldwide against the transgenic plants. So why not to go with the cisgenic approach? I will do the manipulation in the same plant so that at least the environmentalist and the ecologist will not be against to me. So it was a smart steps taken by the scientists, and then they come to the conclusion that go for the genome editing, like edit the genes as per your desire. So say for example for the student, everybody is using this sweet corn. So only few people are knowing that why this sweet corn is so sweet. So what the manipulation has been done, and this company Monsanto who was selling this seed, what they simply do, they down-regulated the one of the gene which is creating the bitterness. And now the same sweet corn from 5 rupees, you are going to the mall, purchasing it for the 50 rupees in the small kuppy, we call in our local slang. So I would just go back. So here, if you look at the genome editing technology, I would give you glimpse that genome editing technology has started long back using the different tools like JFN, TALEN, CRISPR-Cas, recombinering and gene pyramiding. I believe that you have heard about all these three, but only few people are knowing about the recombinering. I will tell you about the recombinering, and that technology was developed by one of my boss, Sabah Kunj, in the Max Planck Institute. I will tell you about the minutes. That's a very fancy technology, and it's very easy to do, and it's cheaper than the CRISPR-Cas. So please go next. So for the students, if you look at the genome editing technology, it looks very cumbersome. कि ये बहुत बड़ी भारी चीज है, बिल्कुल ऐसा नहीं. It's a very easy. You have to understand there is one common feature in any genomic technology, that you have to recognize the sequence where I have to cut and how to press the desired thing and ligate the thing. It is as simple as I am saying. Only thing is that you have to understand what is the precision I have to take and what the molecular tool I have to use, what is present in that cellular organism. So that's all. So if you look at the pathway of genomic technology, it is common in all. So it's like homologous joining, popular way to disrupt the gene, creating a small insertion deletion to the specific point. Say for example, if it is your genome, add the donor DNA, homologous recombination, and you get the target of replacement. Or second, go for the non-homologous gene editing. That here it gives you the freedom that you can put the desired trait at any place as per your. So this is the common pathway which is common in all the genome editing technology. Next please. So the first genome editing technology which was launched, that is called gene finger nucleases, and it was in 2005. Make the story easy, that the gene is present in this DNA binding domain which keep this finger so that it can go and find and fit into the groove. And here in little cartoon you can see that how they recognize the different groove of the DNA, and there is a specific nucleases who cut it. So you can design the gene finger as per your requirement and put a nuclease in front of it, and you can do the editing, cut the DNA at the desired place, and it was used. And the restriction enzyme FOC1 was used for the cutting the double strand break, and this was the first technology come into the existence 2005. Next please. So this is the representation of the same thing, because I am not allowed to speak more than 35 minutes, so I will just skip some slide. Next please. So this is another thing is called as transcription activator like effector nucleases. This is a very important technology. Here the transcription activator like effector is a class of protein with a DNA binding domain, because this is a special protein which has capability to bind a specific region of the DNA. So the TALE protein is a bacterial effector. It was discovered in 2009. And it is naturally occurring TALE repeat comprised of the 20, so it's there only the 10 to 15 repeats that bind to recognize the specific DNA. So if you want to cut the DNA, you have to develop a TALE protein with a nuclease, so you can recognize a specific part of the DNA where you want to do the editing. So that was this concept. Next please. Next please. So then if you look at the existing technology, here JFN, TALEN, and I will talk about the CRISPR-Cas. Next back. So here this slide is very important to show the comparative, because all these TALEN and the JFN have the off-site target. Sometimes if you want to cut the position, it do not recognize it. So that's why they are having the off-site problem. So if you look at this comparison, efficiency target is only 2 to 12%, means it can do the 90% mistake. And here 76% means it's very high, and CRISPR-Cas is the highest, like more than 80%. It means the efficiency rate increases, and that is also a very good sign for the technical advancement. And it takes month JFN, TALEN takes week, and the CRISPR-Cas take only few days. So it is under advancement, means we want to do the things with efficiency and the accuracy. That's the important thing. So next please. Here So why we want to develop the geometric technology? The application to produce the crop resistance and pest disease. We want to produce the tolerant crops, especially for the diabetic stress tolerance. We want to increase the yield. We improve the quality and spread the hybrid breeding, because you know that the classical breeding takes a lot of time. So we want to do the things in the shorter period. And these are certain examples. If you look at till 2016, mainly the Talon was the one of the important tool which has been used for the geometric technology. But after that, you can see the CRISPR-Cas occupy all the places. Please note, move next. So again, I am privileged to show the picture of this lady. I was in Umeå Plant Science Center, Sweden from 2008 to 2013. And I used to take a coffee with the carpenter. And she is such a simple lady, because I was in the common postdoc between the plant physiology and the chemistry department. And she was talking at that time the CRISPR-Cas. And we were not taking seriously in the group meeting and all these things. And at that time, I remember 2012, a talk. She said, one day this technology will bring the Nobel Prize. And guys, you will be surprised that her words become correct, and that she got the Nobel Prize, I think 2 years back. So here, for those who don't know about the CRISPR-Cas technology, I would like to just introduce in 1 or 2 minutes. This is the cluster regulatory interstate short pandemic repeats, was first identified in the 1987. My dear student, one of the important issue what I want to address to you, that we as a biotechnologists, molecular biologists, we are using most of the thing which is available in the nature. We are simply using them in a smart way. Still today, as a human, as a scientist, we have not developed a single technology which is totally man-made. We are getting the things from the nature, manipulating for our own interest. So here, the CRISPR is well known to the world because it's an autoimmune response against the bacteriophages found in the bacteria. So here, 1987 it was reported, and with the help of a nuclease which is called Cas9, I will tell you about this thing. The entire method was developed, that how this is going to recognize and how a nuclease. Again, a recognition point and a cutting point. So this is the timeline, 1987, that the technique was discovered, and till 2012 it was used. And you will be surprised, there was a science paper from Umeå Plant Science Center, Sweden, and where the corresponding author was my supervisor, Professor Stephen Johnson. They developed the first cabbage, CRISPR-Cas cabbage in Sweden. And now the country like Sweden allowed that CRISPR-Cas technology to launch in that particular country. So I'm lucky that at least I have been the part of these people in surroundings who have developed this technology. Next please. So this is called as the genetic Caesar. You can use it for the genetic Caesar. Next please. So next. So here. So what is how it works? Because when the bacteriophage attack to any bacteria, bacteria is so smart, they reserve a part of the DNA and integrate into the main genome, so that when the next time attack will come, it can degrade and attack to that bacteriophage. That is the basic theme behind this technology, and the scientists using this technology, putting a nuclease along with that, and that is known as the CRISPR-Cas. Next please. So this is here, just for audience and for especially for the students. Here you can see, here you have a Cas9 protein and the target RNA, and we can design the RNA specific to that particular region with adding the Cas in that, and we can design the thing and target a particular area of the DNA to do the geometric. Next please. So this is the different mechanism. I am not going to explain. Please move ahead. Move. So here, if you look at the application part, because technology you can read in the books. So if you look at the CRISPR-Cas application, it is used technology that has been used for the site-specific DNA integration, gene expression regulation. Here the genome-wide screening. So one technology is the recombinating. That is also kind of genomic technology, where you can also do the manipulation. Next, please. Next. Gene pyramiding. Nowadays, people are trying to take the trait and try to manipulate that trait and putting into desired species. That is called of gene pyramiding. That is also one of the advancements. Next, please. So now I would like to bring the system biology, large scale biology, and many people are working in that. And the system biology is comprised of the genomics, transcriptomics, proteomics, and the metabolomics. Please next. So omics is the technology where you can do the genomics, transcriptomics, proteomics, metabolomics, lipidomics to understand the plant as a system level, that how the different pathway, different networks are integrated with each other. Move ahead. Next. Here. So this is very important for the students. Here, if you look at the 1953 is the discovery of the structure of the DNA. 1976 there was the Sanger sequencing come. Human genome come into the 2000, and after that 2001 came the first plant genome, and before that the two prokaryotes like Synechocystis 1976, and after that Enabina 1998. Photosynthetic prokaryotes and eukaryotes come to existence, and now if you go to the phytosome, every day a new sequence is coming. From India, we also have the complete genome sequence from the chickpea, tulsi, and a lot of new things are coming. So this is how we are getting the new knowledge. Since I have been instructed that time to be finished faster, so I will not take more than 10 minutes. Please move ahead. So this is NGS. Next generation sequencing platform is very, very important because it offers you the fast sequencing result and it's a cheaper platform. Next, please. So this is the different platform. You isolate the DNA, adapt the primers and do the sequencing. Next. So there are a lot of NGS platform now is automatic like pyrosequencing. So Lexa, Illumina was the first NGS platform. Helicose and different solid is the most recent one. So this is the NGS platform. We can use it for the target therapy, biomarkers, pharmaceuticals, pharmacology and pharmacology. Next. Transcriptomics I can skip. Please move ahead. RNA-seq, move ahead. Please move ahead. Go ahead, go ahead, go ahead. So proteomics, because you know that proteins are the ultimate player. Whenever I give a lecture to the students, I use the one phrase that proteins are the master player in the drama of life. Sukh mein protein, dukh mein protein. Janm mein protein, mrityu mein protein. So proteins are everywhere. So proteomics is the very latest branch of the plant science, the term coined by Australian postdoc in 1985. Here it gives you the idea that what is the protein dynamics is changing. And you know that this is the beautiful molecule. A same protein can do the different function, different conformation in the different state of the life. Next please. So this is the how you do the for the large scale proteomics. The first technology which was evolved, that is the two-dimensional gerophoresis, where you separate the, just hold on, next please, next, where you separate the protein first on the isoelectric point and the second on the mass, next please. So this is the gentleman, O'Farrell, who developed this technology, and I would like to show the first 2D gel. He was not knowing the one gel is going to develop a whole new branch of the life. So next please. So this was the first 2D gel which was published in 1976 where the proteomics has started. Next please. Next. I would like to give one example. My students may understand that how this technology can be useful. So I am giving my own published paper where we compared the Enabina proteome at the 4 sets of temperature 43, 48, 53 and 58. These are the 2D gels. Next please. We found that 13 proteins showing the significant changes over the 2D gel. Next. Next please. Next. Just hold on here. Because generally people know that the heat shock protein are the biggest player in the heat shock response. But by using the 2D, put the drop, put next, enter here. We found that FBPase, FBA, GADPH was the 3 emerged enzyme and ASPC was the key player apart from the heat shark using the proteomics, and we published this in a very good journal, European Journal of Mycology, and we cross-validated data with the real-time PCR. Next, please. Next. Now the metabolomics is another emerging area where people are trying to what is the entire metabolomic changing. Next, please. Next. Here I want to give you one example what I did during my postdoc. So this is the same Arabidopsis plant growing at the 4 light intensity, low light, normal light, and high light and field. When we keep this plant, how they adapt at the metabolome level. Next, please. Next. Here. Look at the metabolic profile of low light, normal light, high light. They are clustered. Put the tab. You can see these metabolome form the three. Next, please. Next. 3 different clusters in the 3 light condition. Next, please. Next. Next. So here, when we try to look at the metabolome changes from 0 to 73 hours in Arabidopsis, click the next, please. Next. So here, back. Here you can see the entire metabolome follow a train, and we publish in Plant and Environment, one of the very good journal in the area of plant sciences. Here you can see how the entire metabolome is changing and follow very specific pattern by using the system biology approach. Next, please. Next. So, next. So, before ending, and thank you to everyone. Emergence of new model system. This is the key thing which I want to give to the audience and all the learned speaker here. Nowadays, we are developing towards the new model system. And sir, with your permission, I just want to take 2 minutes to finish this talk. So here you know that 2008, because this is the Department of Botany, and many people say classical biology don't have a scope. I'm telling you it's a huge huge scope. Because 2008, the first complete genome sequence of a mass from Piscometrella betens, and that is from the Rescue Lab, Germany. So I know him personally. And 2017, because my department of botany is running the MSc Botany from 1919, means so 100 years we have completed, and we are cutting the section of Markensia from 100 years. And you will surprise, the complete genome of Markensia polymorpha was published by a scientist from USA in 2017 in Cell. So 2017 this Piscometrella Markensia polymorpha come, and now the new model system is coming, and that leaves the possibility that as a plant science, and what is the importance, because the bryophytes were the early dwellers from water to land. So if you understand their complexity, you can find many important genes which can be biotechnologically explored. So with these remarks, I am very thanking once again for organizers for giving me opportunity. I hope that my talk will benefit you. I am here and I am welcoming you the question. You can interact me, we can discuss further. Thank you very much.
Thank you so much, sir. May I extend my heartiest gratitude to this August of gathering, the galaxy of high-profile educationalists, proud professors, incredible students. Sometimes it makes you feel how blessed and important you are when you share the stage with some really amazing personalities. Now, I will request our honorable, respected principal, sir, Professor G.C. Behera, to felicitate the honorable guests and the eminent speakers. First of all, I would like to request our principal, sir, for felicitate our chief guest, Professor R.C. Mohanty, sir. Our respected principal, sir, please felicitate Professor R.C. Mohanty, sir, the honorable guest, the chief guest. Now I'd like to request our principal, sir, for felicitate Professor K. Vijay Kumar, former chairman, CSTT New Delhi, Ministry of HRD, Government of India. It's an honor to have him in the stage. A big hand for him. Now I would like to request our principal sir to felicitate our guest of honor, Professor Surya Bihari Satpathy sir, former professor and head teaching department of botany, Utkal University. It's a great honor to have you here, sir. And finally, the eminent speaker, the keynote speaker for the seminar, Dr. Yogesh Mishra sir, Assistant Professor, Department of Botany, Banaras Hindu University. Please, sir, felicitate the honorable eminent speaker, sir.
Now I request Professor Ramchandra Man sir to felicitate our honorable principal, Professor D.C. Beckham.
A big hand for our beloved principal, sir, Mr. Gopal Chandra Behra, sir. Now I will request to Dr. Prabhakar Malik, sir, for a vote of thanks for the seminar. Thank you.
Esteemed principal sir, convener of this meeting, Professor Gopal Chandra Behra sir, honorable chief guest of this seminar, Professor Ramachandra Mahanti, head of the Department of Botany from Utkal University, and revered guest of honor, Professor A. Vijay Kumar, Chairman of CSTI, New Delhi, from University of Israel, and respected our guest, Professor Surya Bihari Satpathy, head of the department, postgraduate department of botany from Utkal University, and respected guest of honor of this seminar, Professor Yogesh Kumar from Banaras Hindu University, and respected organizing secretary, Dr. Gautam Kumar Das, and co-organizing secretary, Dr. Himanshu Shekhar Mahapatra, and respected members of the governing body, Professor Ajay Kumar Samal sir, and Professor Sheikh Samsul sir, and former principal of this institution, Professor Nandakishore Koredal sir, Prasanna Kumar Parijay sir, and invited guest delegates, media personnel, staff members of this institution, and my dear students. It is the proud and privilege for me to offer vote of thanks to the members who are present in this meeting. First of all, I express my sincere gratitude and thanks to our chief guest, Professor Ramachandra Mahanti sir, to the former head of the department, postgraduate department of botany of Utkal University. He has accepted our invitation and also shares his views and suggestions, which has immensely helped us for the academic excellence of this institution. On behalf of this institution, once again, I express my sincere thanks from this institution. On behalf of this institution, I extend my heartfelt thanks to Professor Vijay Kumar sir, the resource person of the seminar from the Ministry of HRD, who has highlighted on the differences between age and syllabus, and he has also given a pertinent idea that in order to conduct research, it does not require the sophisticated instruments. The available resources, taking into consideration the available resources, anybody can conduct the research. Very innovative ideas. Most of the students must aware that the institution does not require the sophisticated items for the conduct of research. From this institution, I also express my gratitude to sir. I also thank and express my gratitude to Professor Kunjo Mihari Satpathy sir, head of the department, former head of the department of Utkal University, who have highlighted very good ideas how the different plants provides us different benefit in different forms, and he has highlighted the different sago and also the potato and vegetables which are immensely help for protecting the different diseases, and how the Tada helpful during the Corona period, and which are remain in surrounding us and provides much more benefit for the society. Sir, we are enlightened and also inspired and also provide thankfulness to your speech, sir. On behalf of the institution, I express my sincere gratitude to Jogesh Mishra sir, with the keynote speaker also, and who have come from long distance from Banaras, and also he has given so many ideas for the benefit of the students and our stakeholders and also our staff members. We are also thankful, sir. I am also thankful to our principal, sir, Professor Gopal Chandra Behera, sir. Although UGC has not funding the amount or the sanction, not sanctioned the money, but he has sanctioned the money out of city fund. So on this auspicious occasion, I also express our gratitude that sir has also sanctioned to conduct the seminar. I am also thankful to the governing body member, Professor Ajay Kumar Samal sir, Samsur sir, those who have consistently guided us to make this institution as an exceptional, as a unique. I also thankful to our invited guests, invited delegates and resource persons, and also the persons who have come from different places, and the members present here. They have also participated in this institution and also patiently listening our speech of the resource person. On this auspicious occasion, I also thank to all the members who are present in this meeting. I also thank to the media persons and also the students, staff, and also the members who are present in this meeting. The meeting could not be successful without the absence of their presence. Thank you all. Thank you all the members of this meeting who are present. Thank you.
Now I would request our Honorable Principal Sir to felicitate former Head of the Department of Botany, Professor Prasanna Kumar Parija sir. Please sir, come on the stage. A big hand for Professor Prasanna Kumar Parija sir. Now I would like to request our Honorable Principal Sir for felicitate former Professor Department of Botany, former head of the Department of Botany, Professor Namita Parijar ma'am. Please ma'am come on the stage. Namita Patra ma'am, sorry. Namita Patra ma'am, please come on the stage. A big hand for Professor Namita Patra ma'am. Thank you, sir. Thank you, everyone.